All right, ladies and gentlemen, Mr. Mannequin and I are going to give you some notes on meiosis. Make sure you have your notes open on a separate tab, and anytime that I'm going too fast, just pause my screencast so that you can have a chance to write in your notes. All right, so what is meiosis? We're comparing meiosis to mitosis. Mitosis was cell division that produced two cells that were identical. Meiosis is cell division that produces, four cells, and the four cells are unique. They're your gametes. So it's cell division that occurs twice to produce four sex cells called the gametes, sperm, egg, or pollen, and they have half the original number of chromosomes. Let's learn more. All right, meiosis is the process that makes sperm, egg, and pollen. And sperm, egg, and pollen in plants carry half the DNA. That way, when they come together, they produce a baby that has the right amount of DNA, called a zygote. So on your notes, you're supposed to write in the word zygote. So when sperm and egg join, a single cell baby called a zygote with the correct amount of DNA is produced. The zygote then begins reproducing rapidly through the process of mitosis. It was meiosis that made the sperm and egg, mitosis that allows the baby to grow. All right, so you should have a diagram. You should have taken a photograph of this in class, but this is the diagram. This is the process of meiosis. Meiosis looks a lot like mitosis. Instead of the phrase I PMAT, we're going to say the phrase I PMAT, PMAT. Notice that interphase doesn't happen twice. It's just at the very beginning. So these are the things that make meiosis different, and then we'll talk about how it's similar to mitosis. First of all, during prophase 1, which is the blue cell, homologous chromosomes find their partners. Now in mitosis, they didn't find their partners. They just were scattered randomly all over the cell. This time they're going to find their homologous partners. And the process of finding their partners is called synapsis. And synapsis happens specifically in meiosis during prophase 1. The reason that synapsis is important is that this is when the chromosomes do that crossing over business, and you can see the diagram in the bottom left. Crossing over is when they're able to trade pieces of DNA. Mother's chromosome trades a section with father's chromosome, so that you're creating chromosomes that have never existed before. All right, another difference is in the very next cell in metaphase 1. In metaphase 1, the chromosomes line up in matched or homologous pairs. In metaphase of mitosis, they lined up single file. In metaphase 1 of meiosis, they line up side by side. And then in the green cell right below that, which is anaphase 1, the cell pulls the partners as bivalent chromosomes. They don't get separated. They separate from their partners to the opposite sides of the cell, so that when the cell divides through telophase, it cuts the chromosome number in half. The rest of meiosis looks just like mitosis. So if you notice in the purple cell, there are now 3 chromosomes because the chromosome number got cut in half. They line up then in the pink cell in the middle. They get pulled apart, anaphase, in the green cell, and telophase again. Remember that the 4 cells at the bottom, those 4 red cells, are the gametes. So those represent 4 sperm, 4 egg, or 4 pollen. It has half the DNA. All right, let's talk about a couple other important concepts that have to do with meiosis. Why is meiosis important? Who cares about it? Well, it has to do with sexual reproduction, and that's really important, and that's what we're going to talk about. So independent assortment is an idea that Mendel came up with. We talked about it once before. We're going to go into more detail here. The 23 pairs of chromosomes that humans have, and this happens in everybody else who reproduces sexually, the 23 pairs of chromosomes line up side by side before the cell divides. That's in metaphase 1. The order in which they line up, though, is a random process. So they can line up on either the left or the right side, sometimes left, sometimes the right. Mathematically, that means that, at least for humans with 23 chromosomes, 23 pairs of chromosomes, there are 2 to the 23rd possible combinations of chromosomes. That's more than 8 million unique combinations of chromosomes for just one sperm or egg, possible combinations. So that's adding a lot to genetic diversity, and we'll get to that, why that matters in just a minute. So what is genetic recombination? Genetic recombination is any event that leads to new combinations of genetic material, and there are 3 circumstances that we're going to talk about primarily. The first one is crossing over. We've mentioned it before. We're mentioning it again. Crossing over is when homologous chromosomes trade little sections of DNA. This creates chromosomes that have never existed before. So again, that's an event that's leading to new combinations of genetic material. The second way we get genetic recombination is through mutations. Although usually mutations are harmless or harmful, and they're not usually beneficial, they still create new combinations of DNA and increase the diversity in our population, any sexually reproducing population. And then finally, random fertilization. We never know which sperm and which egg are going to come together. There are about 70 trillion possible combinations of chromosomes in the zygote. So that means that each individual, you in this case, are very, very unique. So why does it matter? Why do we care about all this genetic recombination? Why is sexual reproduction advantageous? Well, we're going to talk about that. Asexual reproduction, the prefix a means without. So asexual reproduction means to reproduce without having sex. What? Who reproduces without having sex? Bacteria do. Bacteria are able to just clone themselves. They split in half. The two babies that come out, so to speak, are identical to the original parent. So they're cloning themselves. That's asexual reproduction. Actually, the strawberry plant down at the bottom can reproduce sexually or asexually. The runner plants are just clones of the original parent plant. So that's an example of asexual reproduction. Strawberries also produce flowers that can be pollinated by things like bees and butterflies, and that's sexual reproduction. So strawberries can do it either way. So what are the advantages? Why do some organisms produce asexually? Why does that even exist on our planet? Well, it turns out that it's really, really efficient. Reproducing asexually, there's no need to waste energy and time finding a mate, and reproduction is able to happen much, much faster. Well, if it's so quick, why do we reproduce sexually? Well, the disadvantage to asexual reproduction is that there's no genetic variation. Except for occasional mutations, the offspring are clones of the parent. The reason we care about that is that if the environment changes and everybody's a clone, there may be no individuals with the needed traits. That population is at risk for going extinct when everybody's a clone. However, with sexual reproduction, which includes things, the meiosis, the mating, and then the fusion of the gametes, we have certain advantages. Sexual reproduction leads to a lot of genetic variation from the three things we just mentioned: crossing over, mutations, and random fertilization of gametes. With sexual reproduction, if the environment changes, because there's this huge diversity in the species, there might be individuals who actually have the needed traits to respond to the environmental changes, and that makes the population less likely to go extinct. That doesn't mean the population won't go extinct. It just gives them a little bit more diversity to handle an event in the environment. So disadvantage to sexual reproduction is that it requires much more time and energy to locate a mate, and so reproduction happens much more slowly. Bacteria can clone themselves every 20 minutes. Human beings can only reproduce, a few times in their lifetimes. So sexual reproduction increases genetic variation, but decreases the number of times that organisms can be reproduced. Finally, we'll just take a look at a comparison between mitosis and meiosis. It cut out several of the steps, so that's not the only cells. But just notice in mitosis, in prophase, they're scattered all around, whereas in meiosis, in prophase 1, they found their partner. So they're sitting side by side with their partner. They are crossing over, so they're trading sections of DNA, creating these unique genetic combinations. In metaphase of mitosis, so I'm going back and forth, in metaphase of mitosis, you see them lined up in the middle, single file, but in metaphase 1 of meiosis, you see them standing side by side with their partner, so that when the cell divides, in meiosis, half the chromosomes go to the left, half the chromosomes go to the right, which means you cut the chromosome number in half. So the ultimate cells at the very end have half the DNA, whereas the cells at the end of mitosis have the same amount of DNA that the original parent cell had. All right, that's it. Let me know if you have any questions.